sharing knowledge and experience is the key to accelerate development of genomic medicine, which is a new field, with a shared goal of advancing genomic medicine in Asia Pacific region. The Asia Pacific Society of Human Genetics and the Hong Kong Genome Institute has decided to collaborate to organize and to co-host this HKGI-APSHG Distinguished Scholar Series. In this series, we bring together renowned clinicians, scientists, and researchers to facilitate international knowledge exchange. My name is Brian Chung. I'm the Chief Medical and Scientific Officer of the Hong Kong Genome Institute, and I am also the President of the Asia Pacific Society of Human Genetics. It gives me great pleasure to introduce our speaker today, Professor Haiyan Chow. Let me introduce Professor Chow. Professor Chow is a professor of genetic medicine at University College London and a UK Heritage Rare Disease Scholar. Her research specializes in the preclinical development and clinical translation of nucleic acid therapies for rare genetic disorders. She serves as the deputy theme lead for the novel therapy theme at the NIHR Biomedical Research Center at UCL Great Ormond Street Hospital, where she leads the individualized RNA therapy program for pediatric rare diseases. Professor Chow is the director of the UK platform of nucleic acid therapy for rare diseases, a specialized initiative supported by the UK Medical Research Council and the NIHR. As the principal investigator on a range of research projects, she is at the forefront of developing RNA-based therapeutics for a wide spectrum of genetic disorders. In addition to her research, Professor Chow directs the MSc in a program in personalized medicine and novel therapies at UCL, where she plays a key role in training the next generation scientists in cutting-edge therapeutic development. Professor Zhou is a member of the World Muscle Society, the Oligonucleotide Therapeutic Society, and the American Society of Gene and Cell Therapy. She is also the UK representative and management committee member of the European Cooperations in Science and Technologies on delivery of antisense RNA therapeutics. We would very much like to listen to your lecture, Professor Chow. Thank you very much for giving us this important lecture.
Thank you very much, Dr. Chung, and thank you for the invitation. And it's such a great pleasure to be here to share with everyone what we have learned from RNA therapy for rare disease. We have been working in this area for many years, and I'm going to start. Just a bit background. Actually, Dr. Chung already introduced. I'm also a Chinese. I'm from China 20 years ago, moved to the UK. I did my medical training in Beijing Union Medical College and then moved to Switzerland for some medical training. After that, I was so obsessed with genetics, and that's in early 2000 and decided to move to the UK. And since then, I worked at Imperial College and then did my postdoc at UCL and then later established my group at UCL and specific on RNA therapy. So today's talk is about the personalized nucleic acid therapy. I would like to share with everyone what we have done, what we have learned, and what's the newest development in this area and what we can do for the future. This is a summary slide of how many FDA-approved antisense drugs currently in market. So the first drug started in 1998, that's formimersen. And you can see that now there are 18 FDA-approved oligonucleotide drug that include antisense and also siRNA. And I also highlight them in different colors. That's for different modality, including splice switching antisense, gene silencing antisense, and also siRNA. So that's around 18 FDA-approved drugs. I didn't put the personalized drug here, but we'll talk about that later on. And you can also see in the most of the drugs that developed in the last 10 years, since the first DMD drug, SMA drug approved. So in the last 10 years, the reason why is because rapid development in technology and also genetic diagnosis that benefit the area development. So the background information about here we focus on antisense drug. So the mechanism of action, and as we know, antisense drugs, they are short DNA or RNA fragment around the 20 base pairs. So it's single nucleotide, and this single nucleotide will bind to our target gene and either will silence the target gene or will modify the gene splicing. We call this splice switching antisense, and also some other strategies can use to upregulate the genes, such as here is a targeted gene upregulation. I will have a brief touch later on. So this is the two major mechanisms of action that focus on gene silencing and splice switching. So for gene silencing antisense, this is a basic design. It's a gapmer design, and gapmer is a mixture of DNA and RNA. So when we design this, we have the DNA in the middle. We call this DNA gap, and with RNAs at each end, we call RNase ring. So the RNase is to reduce the toxicity and increase the binding affinity, and also increase the specificity. Actually, it's in the middle, the DNA phosphoryside DNA part, and bind to our target mRNA, activates an endogenous RNase H and make the gene degraded. So that's how gapmer antisense work. And there are two major applications for gapmer antisense. One is general gene silencing. Another is allele-specific silencing. As you can see, for allele-specific silencing, so it's mainly used for gain of function mutation when the mutation causes toxic effect. There's again dominant negative effect or toxic effect. We will apply allele-specific silencing. But for the general silencing, we usually use to interrupt some, for example, a metabolic cascade, a pathway, and you don't target the mutation or the mutant gene itself. Instead, you can target the upstream of the pathway and stop the pathway. So that's how why many of the FDA approved ASO or siRNA drug as they work in that way. And compared to siRNA, here antisense has broader targeting flexibility. As we know, siRNA only targets the coding region, the exons. But for antisense, it can bind to exons, can bind to intron, can bind to 5' UTRs, and to block transcription and translation. So this is the background information about gene silencing antisense using gapmer. And here you can see about 13 out of 18 the FDA approved drug, they are on gene silencing approach. So I'll highlight them in blue. And next I'll give some example. The most exciting example for allele because general silencing, I'll skip that one. You can see from all these the 13 FDA approved drug are mainly focused on the allele specific silencing, which is relatively new compared to general silencing. One exciting example is the KIF1A antisense. They published this. It's from Chung's lab in Boston Children's Hospital. They work with Enlorum and published last year in Nature Medicine. For that condition, they developed this antisense target a common SNP, and that SNP is incised with a mutation. So by binding to this SNP and then silence that allele, and because the mutation sits on the same allele, and then we can also silence the mutant transcript. And they show in that paper, they show the patient respond very well. And now we also collaborate with Enlorum, with UpNet. So we try to import that antisense to see if we can benefit our UK patient. And the one thing I would like to mention to check if the target mutation and the target SNP, if they are incised on the same allele, we need to use long-read DNA sequencing here. And we use the UCL surveys from Queen Square Institute of Neurology. And we already identified one UK patient suitable, amenable for this ASO development environment. Hopefully, we will get some good news for the family. Another case about allele-specific silencing is that's a few years ago in my lab we developed the allele-specific silencing for collagen 6 related congenital muscular dystrophy, and that CMD can be caused by either dominant or recessive mutation. So we mainly focus on dominant mutation, which account for almost all the uh, based on muscular dystrophy, 95 based on and 50% of Ullrich muscular dystrophy are due to dominant mutation. So we think that this will be a very useful approach. So in that case, in this study, we confirm that allele-specific gap antisense, if we compare the efficacy by targeting either pre-messenger RNA or mRNA, and we found ASO show more silencing efficacy when target mRNA instead of pre-messenger RNA. So that's quite important information to guide our design when we should focus on exonic sequence or intronic sequence. And then later on, just the last week, we worked with the Oxford team and they developed the AI called the Oligo AI. When they analyze all the FDA approved or clinical trial and gene silencing ASO clinical trial and from FDA database, and they found the best candidate actually should sit in the 3 UTR. That's where most success. That's also some hint for ASO design, especially earlier specific one. And then after that, we further improve this approach. And so because allele specific silencing, we usually target the single nucleotide chain, which is quite challenging and needs a lot of improvement to enhance the specificity. At the same time, you don't want your design affect the efficiency as well. So in this paper we published last year, so we show that by changing from the basic GAPMET design to mixed-MET design, and this is some illustration to show how we can design mixed-MET. And mixed-MET design can increase the specificity by threefold. If we further introduce some mismatches to our antisense sequence, and this, for example, here we introduce different mismatches, and mismatches mixed antisense design can increase the specificity by tenfold. At the same time, they don't affect the efficacy. So this, and also we show the importance of structure modification in ASO design that also affects the efficacy and the specificity for allele specific. So this guideline, we got lots of response and really improve or enhance the design of allele specific antisense. So based on that, because this is all in vitro, we test in patients' cell line. And then later on, we move to in vivo, as we know. So in vitro work doesn't mean in vivo work, and in vivo is usually more challenging than in vitro. So very pleased to update everyone. So this is in another disease model called sensory neuropathy, and the patient carry UK founder mutation. But in this study, we use a different mouse model as surrogate. In this surrogate mouse model, which also carry a dominant mutation, so we design mouse ASO target that dominant mutation and to see how it works in vivo. And it's because the disease model is sensory neuropathy, so the target organs include the liver, dorsal root ganglia, and the sciatic nerve. So this is one of the study where after weekly subcutaneous injection for 8 weeks, you see that our antisense can specifically silence the mutant transcript more than 90% in liver, more than 50% in dorsal root ganglia, as well as neuron C, and also certain degree of silencing in sciatic nerve usually, which is quite hard to target. So this paper has just been published a brain actually just online yesterday. So if you're interested, you could have a good look at that one. And so apart from now, we moved from allele-specific silencing or gene silencing approach to splice switching approach. So the two splice switching approach, one is exon skipping, another one is exon inclusion, just opposite. Exon inclusion is one exon, usually it doesn't work, and you want to here in mature mRNA, you don't see it, but with you want to rescue it and get it back. So the classic example is exon skipping antisense in spinal muscular atrophy. As we know, spinal muscular atrophy is one of the common rare disease with a prevalence of 1 in 6,000 to 10,000 general population, and it's a recessive. It affects the motor neuron, cause motor neuron death, and then followed by muscular atrophy. And then genetic defect of SMA is due to a large genomic deletion in around exon 7 region. And so apart from SMA, so the SMA patient, they lose the function of SMN1. So for all of us, apart from SMN1 gene, we also have SMN2 genes. However, due to a C2T single nucleotide change in exon 7, and the exon 7 in SMN2 gene cannot be properly transcribed. And then with the product lack of exon 7, and which is not functional. So for patients who has loss of function in SMN1, they still have SMN2. There's nothing wrong with SMN2, but SMN2 is not functional. So what we did is we designed ASO antisense approach to rescue exon 7, do exon 7 inclusion, get it back, and make it full length and become fully functional. So that's how nusinersen works. And then, so that's because I started this project in 2008, many years ago. So have lots of paper publications in this area. So after we found, as people found the function of exon 7, the importance of exon 7 in the splicing of SMN2, many studied around it to check which area, which domain affect exon 7 splicing. So this is all the publications, and then based on that, different group also design different strategies targeting exon 7, correct the splicing of exon 7, try to rescue the function of SMN. And based on that, our lab, we mainly focus on two areas. Our first project is working on the 6 nucleotide. Remember the one C2T change sitting here? We focus on that region, and we design this bifunctional antisense. And so this is the tailed one part of the antisense bind to the C2T change, another exon 7, and with a tail to recruit the splicing factor SF2ASF. So we call this bifunctional antisense design. It worked very well in vitro. However, it didn't work in vivo. So that one we kind of dropped. And then later on, we moved to the intronic region. That's intronic splicing silencer region. Reported by Sin in 2006. That's also the region where Adrian Kriener's group and Ionis they focus on. So they focus on MOE chemistry. We focus on morpholino backbone, and we share that one work beautifully. And really we got very consistent data as what Adrian's team published and Ionis team published. So at the end, the 18 MOE antisense called nusinersen. They moved to clinical trial, and now that's the treatment is patient receives the intrathecal injection, and it includes two stages. One is the loading phase. For loading phase, so in the first 2 months, patient will receive 4 injections. So every 2 weeks for the first month, and then wait for 1 month and the fourth injection. After that, every 4 months and for life. Patient responds very well to this treatment, and currently there are around 20,000 patients worldwide they're receiving the treatment. At the same time, there's also, of course, there's the small molecular called risdiplam and gene therapy called Zolgensma. All these three drugs work quite well in asthma patients. And of course, in the trial starts in severe type 1 SMA patient. Now the drug can be used any type of SMA. And from the severe, and now also in the UK and many other countries, they start neonatal asthma try to push SMA include in newborn screening and can start as soon as possible if they found the gene that is a mutation genetic defect. In some group and Richard Finger's group, they also start prenatal treatment to see if they can further improve the clinical efficacy. So this are removed rapidly. So another way, and then we moved from exon inclusion to exon skipping. So I'm sure immediately you will think Duchenne muscular dystrophy. That's the best case example. So for exon skipping, there are two strategies we want to apply to. One is to rescue the reading frame. That's what we did in Duchenne. In Duchenne, there is a nonsense mutation in one of the gene, usually sitting in the middle, and they will affect the reading frame and create a premature terminating code. And with exon, with antisense, we can get rid of that exon and rescue the reading frame. So make the product, although it's truncated, but it's functional. Another strategy we use is for deep intronic mutation. Usually those mutations will cause intronic retention, or we call pseudoexon. So we can use exon skipping to get rid of that pseudoexon and correct the reading frame. So that's the two strategy we use. We use exon skipping. And so that's the example for Duchenne muscular dystrophy. That's one example with large deletion from exon 48 to 50. And if we skip exon 51, we can restore the reading frame. And currently, there are 4 antisense drugs have been approved by FDA, although they're not approved by by EMA, but still they're in market. So that's ertapenem for exon 51 skipping, and golodirsen 53, and also for 45. So currently they focus on exon 51 skipping, 53 skipping, and exon 45 skipping. So this approach, they are mutation specific. And so I would like to give some example from still in preclinical development stage. So one is still back to collagen 6 related congenital muscular dystrophy. So this deep intronic mutation, it takes clinical genetics quite a while to find out the mutation, because from clinic, a group of patients, they definitely show the clinical features of congenital muscular dystrophy and exactly point to collagen 6. But by whole genome sequence, they couldn't find any mutation. It's only by RNA, later on by RNA sequencing in muscle, and they found this strange abnormal transcript, and then later on identified that it's a dipintronic mutation sitting there. And after that, we designed the ASO in collaboration with Custom Bonneman's lab in NIH. So they work on PMO backbone, we work on MOE backbone. So we designed ASO targeting this pseudoexon, or pseudoexon 11, sitting in exon 11 and 12. So that product. So targeting that and induce pseudoexon skipping, and then we showed the ASO approach worked beautifully. So that paper published in here. This is a paper, exon skipping paper. So from my lab. And then regarding exon skipping, that's a very powerful approach being used in personalized treatment for ultra-rare disease. So the first personalized antisense therapy is developed by Professor Tim Yu's lab in the Boston Children's Hospital, and for a girl called Mila. I think many of you already heard the story of Mila and also the development of Millicent. So the mutation is in CLNN7, also called MFSD8. It's a deep intronic one, and then causes intronic retention. And ASO is designed targets this area. And the Milosen, the drug developed in a year. That's a few years ago in 2018. Since Mila was diagnosed and they found the genetic defect, and teams lab they work rapidly and design is work with MHR, and in less than a year they started treatment for Mila. Although at the first 6 months, Mila responded very well to the drug, but her condition at that time is quite late already, and after 6 months her condition declined, and 2 years after the treatment she still passed away. But that also remind us it's quite important to start the intervention as early as possible, especially for these degenerative conditions. Talking about now we move to the personalized medicine area, and this is after Millicent and Team's Lab and many other groups, including Lorem, they start this individualized antisense therapy for different conditions. So this is I only put some example here, but you can find all the list. Actually, there's a lovely table with all the individualized is developed by different group in different country. So this is a paper. So you can find, and there's a table summarize all the individualized ASO from different lab, including Learn. So one ASO developed by Teams Group is a gene silencing GAPMA ASO for KCNT1 infant epilepsy. So here the name is always use the first patient's name, so the first baby called Valeria. So she got this ASO treatment. Unfortunately, later on, 6 months later, she shows very very severe side effects. It's the uh, uh, hydrocephalus. So the trial has been stopped, but Valeria passed away. The first patient died, and the second patient also showed the same side effect, hydrocephalus. So the drug stopped, and so that also remind the area to be more cautious for this individualized ASO therapy development. And after that, if FDA respond very rapidly and they publish this guideline for individualized antisense therapy, all the criteria or the guidance for uh, investigators to follow. That's very helpful. Now we still use, including a Lorem industry and also charity and different academic lab. We follow this guidance. After that, this is another antisense also target a deep introntic mutation for two siblings affected caused by mutation in CLN3. This is also a mutation specific approach. And Leucin, that's interesting because most ASO here, they are in MOE, it's a MOE backbone. But here they use the block nucleic acid backbone. That backbone before the first or only clinical trial of LNA antisense failed many years ago when they used LNA ASO to target a liver disease, but this shows the kidney toxicity. So it's kind of all the pharmaceutical companies are quite cautious to use LNA. But here the patient received LNA by intrathecal injection. Some patients tolerate it very well and definitely improves the clinical features. So it worked in Leo. And then another one is I would like to mention is the one for ataxia. So the initial ASO developed in Teams Lab, and it shows the very promising clinical efficacy. And now the same ASO have been adopted by the German group and now treated in Germany for more patients. So that's a successful case from end of one to end of many. Another very successful case is Jackie Fusin. So the story is start from the a girl called Jackie, and she is diagnosed with mutation in fuse gene and with ALS. She already has a sibling died of this mutation. So once she's diagnosed, and immediately the charity started the develop of uh, the antisense, and Laura support this, and then Jackie got the treatment. Actually, at that time, and Laura they have some ASO, and so Jackie respond very well to this treatment. And then from this N of one, now it moved. Ionis moved this ASO now has another name, Wolf Nursing, and this drug now moved to phase 3 trial. So that's a successful case of from N of one to a large population phase 3 trial. That's also what for many rare disease. So our the drug developed may not just benefit a single patient. Instead, later you will realize it can benefit more patients instead of N of one. So in the UK, we also have we also have the program for customized antisense therapy. We call the CAT program, and funded by NIH, Ghosh Biomedical Research Center, led by myself. And then, so this is a mutation we target, a stipindronic mutation in MPC1 gene. The patient carries combined heterozygous mutation. One is a missense mutation on the paternal allele, another is stipindronic mutation in the maternal allele. And the second mutation does take a while to be identified, and that's from the 100,000 Genome Project. So after we identified the dipindronic mutation, at my lab, we designed ASO and target this area. And so here is some data. We show the ASO work by correcting the pseudoexon and get rid of pseudoexon and change it back to here. This is the wild type normal transcript, and the top is the one with mutation with pseudoexon. So we show our ASO work very well in patients' fibroblasts at RNA level, and later on we also show this antisense can restore protein. So here, this is the in patient without treatment, this MPC1 protein reduced, and then after treatment, you can see protein increase to not to normal level, but at least 50% level. So that we because this is the recessive conditions, we think 50% should be functional. And also at the functional level, we measured the MPC1 protein and also some the sphingolipid by mass spectrometry, and to show the treatment can also restore those biomarkers. That is the in cultured cells. And then this project and the ASO then later on being adopted by Everyone Medicine, which is a company focused on N of one treatment, founded by Mila's mom, Julia Vettarello, and also teams group. And then the company has expanded a lot. So they focus on the UK population, and so MPC1 is the first case they have accepted, and now the clinical trial authorization application has been approved by MHRA last month, and with a master protocol. So we will start very excited. We will start the first clinical trial in January 2026. So the antisense we call it Evasen, because the girl's name is Eva. And then with this master protocol, we hope we can include more patients, also with different mutations, but they all have the neurodegenerative conditions can be included in this protocol and get a faster approval from MHRA. And also the plan is in the future, this program can be integrated or accepted by NHS system, for example, approved by NHS England, get reimbursement from NHS to continue to with government support to for the maintenance of this program to support get the refund and then use it to support more ASO development for more patients. So based on that, in 2023, we established the UK platform for nucleic acid therapy for rare disease, short for APNet. So at that time, we feel there is a strong need. So we have all the technology from different groups. We work on different modality, different chemistry, different model system. And our clinical team, they are wonderful, the world-leading clinical team. We have the unique patient population for rare disease. We have wonderful gel genomic inland 100,000 genome data and many patients before the undiagnosed patient, now they have genetic diagnosis. And what's the next? After the genetic diagnosis, what are we going to do? We have everything here. We have, all the technology needed for ASO development or nucleic acid therapy development. We do need an organization to put everyone together, all the stakeholders together, and to promote the clinical translation or preclinical development and clinical translation. So this is the aim of APNet. So this is the management team of APNet. It's based at UCL, led by me, and the Professor Montone is my deputy, and then with a group of co-PIs from Cambridge, Oxford, NATA, and also Genomic England. And we also collaborate with 5 HR medical research centers in Oxford, Cambridge, and London as well. And that's our aim, to put all the stakeholders together and to provide a clear roadmap for rapid bench to bedside translation of nucleic acid therapy for a disease. And we mainly focus on 3 areas. One is target selection. That's a group working on the guideline, the AFNet guideline. We also have the preclinical development group, and that's, for example, where NPC1 antisense developed. That's from this group, and also clinical translation. And then once we have all this developed, and we have a group work with the team, the company, and also with MHI to have, especially now, to have a clear guidance in the UK for nucleic acid therapy for rare disease. Currently, there is no such guidance exists. We still use FDA's guideline. We also work very closely with N of1 Collaborative, and we contribute to this, as many of you already know, this N1C guideline. And then we work with them, and also now we would like to have a UK-based guideline or AFNet guideline, where we'll focus on the N1C guideline, mainly focus on the different genetic variants. We will focus on not just genetic variants, we also need to consider the condition, what disease it is. And if because that's about the delivery, about distribution. Some organs difficult to target by ASL, some are easy. For example, by intrathecal injection, we can target the brain, and liver is always as the best target for ASL. Will 50% ASL will get there. And muscle, there's some ASL approved for DMD, but the clinical efficacy is still challenging to achieve. So we include here, but we do need the new strategy to improve muscle delivery, and also eye. That's another treatable organ. So we focus on these 4 organs, but also we have a list of different other organs to target, and also functional model. If we need to consider if the gene function is well known, if any model system available, for example, patient's material, fibroblast, iPS cell, or iPS differential different model system, if they are available, if there's any mouse model available. So all these things we need to consider. And also another one is patient, if they are suitable for treatment, if the disease is too severe, because the develop ASL will take time, usually take around 2 years. If the patient is still suitable for treatment in 2 years' time, or if it's too late to treat it, of course, the earlier the best. So that's what we need to consider. And then, so our work has been included in early this year, from UK government, the England Rare Disease Action Plan to 2025. So this is the in Action 39 that mentions the individualized therapy, and the aim of that is to develop a national operating framework for individualized therapy. And here, particularly they mention antisense, and then also for to consider for long term, this treatment can be uh, accepted by NHS. And in that action plan, and we also now we work with Rare Therapy Launchpad and also with NATA as well. I think now we mentioned the gene silencing approach and specificity approach, antisense approach. Another one I haven't mentioned is the targeted gene upregulation, or called TANGO. So that one has been uh, you will see that currently there is no FDA approved drug being developed. It's still at uh, that approach is less mature than gene silencing and splice switching. Still need lots of preclinical development and also some on clinical trial. For example, the one targeting the poison exon skipping, the Dravet syndrome, that's the ASO approach they use. And the one by blocking the naturally occurring antisense transcript, that's the approach they use for, but you designed ASO to target a long non-coding RNA, which cause the paternal allele imprinting. So by blocking that long non-coding RNA, we can release the imprinting as a paternal back. That's mainly for conditions of hyperinsufficiency. So that's what that's the two most promising cases in clinical trial. The others, for example, the open open reading frame is still at early preclinical development stage. There's no clinical trial yet. So still, we still need new strategies for gene upregulation. And then some background information about antisense toxicity. It's a very powerful technology, but at the same time, there is also limitation. Unlike any other drug, the ASO drug also have toxicity, and this toxicity can be from two sides, or we call this twofold toxicity. One is from the technology itself, it's platform technology toxicity associated with what chemistry we use, what backbone we use, what kind of conjugation we use. And also another one is the antisense sequence itself. This is sequence dependent talks. So that part we could exclude by in silico screening to check the off target, and also by RNA sequencing to check off target. Also, some different specific nucleotide combination, for example, CG usually cause inflammation, and also we always need to avoid, don't put the G at 3' side of your ASO. That will cause chronic neurotoxicity. So this is the kind of tips when we do the ASO design. The general toxicity that include immunostimulation, inflammation, complement activation, and also the liver and kidney toxicity always need to check, and also blood, for example, the platelet number reduced, and also coagulation. So that's the blood. And then central nervous system, that's mainly for intrathecal injection. For example, Huntington disease, the ASO failed in phase 3 trial. That's because the chronic neurotoxicity. And also another one, the injection site reaction. That's mainly linked to the first antisense trial in Duchenne muscular dystrophy, when they use two methyl backbone, and that's quite backbone associated. And those patients, even by themselves, they can figure out if they are in the placebo control group or treated group, because from the injection site, if they have a strong reaction site skin reaction, and they know they are in the treatment group, those patients still, and the injection side reaction, they still suffer after so many years. They still suffer the side effect from the injection side reaction. reaction and the other flu-like. This is very common. And then, so this is a nice paper for the preclinical safety assessment we can follow. It's a methodology paper, very, very detailed and comprehensive. And also FDA last year, they published this non-clinical safety assessment for oligonucleotide we can cite or refer to. Another key parameters for ASO development is the delivery and biodistribution. And we know it's for brain. That's the reason why people like brain, because we can do intrathecal injection. Also, they like kidney. No, sorry, they like liver because 50% of drug will stay in liver, and then for kidney, most of they've been cleaned by kidney. But if you think liver is nice target, actually it's wrong. Even the drug ASO can go to liver, but they will not be uptaken by hepatocyte. Most of the drug will be uptaken by Kupffer cell, that's macrophage cell in liver. So to enhance liver delivery, still the people develop this gamma conjugation. Gamma is a ligand for the ASPGR receptor, exclusively expressed on the surface of hepatocyte, and there are around 20,000 copies of the receptor on each hepatocyte, and then the gamma would conjugate our ASO, and then the gamma, the ligand, will bind to the receptor, and then which will get this hepatocyte specific delivery. Now, gamma conjugated antisense has, the clinical trial showed very promising data. For example, this is for HTDR-related amyloidosis. There are two approaches. One is antisense called inotersen. Another approach is siRNA called patisiran. So these two approach, if it's unconjugated, of course, patisiran is in LNP formulation, but inotersen is unconjugated. So this is the treatment regimen. For inotersen, 300 mg per week, weekly subcutaneous injection, and siRNA is the lower one, but still need every 3 weeks. By conjugating them with GalNAc, now there is a new version of inotersen called eplontersen. So we can dramatically reduce the dose and also the frequency from weekly to monthly. For siRNA, it even works better, although similar, but the frequency can increase from 3 weeks to every 3 months. So this is a dramatic improve and also can make a patient's life much easier. You don't need to go to hospital every week. You just every month you do the injection. So more accept, more convenient for patient, and also the dramatically reduce those clinical side effects. And that's the reason the FDA approved drug in the last 5 years, all the metabolic drug, if the target is the liver, they all are, so I highlight in red, they are gamma conjugated. So you see how popular that conjugation is. For muscle targeting here, we now have transferrin receptor antibody conjugated ASO. So that's a new approach. And from clinical trial, it worked beautifully. So the trial performed by Avidity and also Dyne. So all shows in Duchenne, it shows a dramatic improve for Duchenne muscular dystrophy and for myotonic dystrophy. So hopefully the drug can be approved by FDA, and then the conjugation can be used in more neuromuscular conditions. And also the Ionis group, they also found actually transferring antibody conjugated ASO can cross blood-brain barrier. That's a big breakthrough, can really change the progress of this area. So now I think this is one of the last slides I would like to show. And this is a traditional drug development. It will take at least 10 to 20 years in the past. For example, the antibody drug and small molecular drug. So that's a traditional way. But for the individualized therapy, especially for antisense or other gene therapy, CAR T therapy, the length can be dramatically reduced to around 2 years. So there is great potential for individualized therapy, and also lots of support from regulators and to promote this. And then the same time, we could start from the individualized therapy, like Jackie Folsom. So from single patient and now move to large phase 2 trial. So that's another way around to develop ASO drug. So I would like to finish to summarize what's today's talk. So you can see nucleic acid therapy is promising for genetic disorders, especially for rare disease, and we expect more antisense drug being approved in the near future, and also more drug being approved by regulators for personalized treatment. We think this approach is it's good, but it's not perfect. There's still those things we need to improve. For example, we need a new chemistry, more efficient and less toxic, new delivery system to enhance the in vivo delivery and enhance the clinical efficacy, and also new mechanism of action. Not just gene silencing, specific gene. We want to target more mutations and, for example, gene upregulation and then other mechanism of action as well. And also, we do need support, for example, regulator approval, a clear system. Uh, for example, in the UK, we work with MHRA now. The MHRA recognize our need and they organize this work stream. And so we draw UpNet now joined this work stream to work on the UK guideline. And also, we nowadays lots of groups start using AI for ASO design. That's actually AI is a brilliant tool for ASO design, and then that can not just design and also find the suitable target. So AI technology, we believe, can dramatically improve the drug development and the dramatically shorten the drug development period. And so this is something we also focus on as well. Yeah. So I would like to stop my talk here and by acknowledging this is the UpNet network we have established globally. I would really like open the collaboration and with our team in Hong Kong in China, and I'm also a Chinese, and our UpNet team and also my lab here.
Thank you, Professor Chow, for this excellent talk. We have learned a lot, and we are very happy with your last statement that you are open to collaboration in Hong Kong, China, and Asia Pacific region, because we do have a lot to learn from you and your team in the UK. I always have the privilege of asking a few first questions. Uh, I'm a clinician. I'm working with patients with rare genetic disorders as well. Now, we are so happy that there are much more opportunities of treatment and therapeutics. So let me start by asking you a more general question. Now, choosing between RNA-based therapy and other potential approaches for N of 1 diseases like CRISPR editing, may I ask, do you have a general approach in deciding which one would be more useful in a certain patient or a certain type of disease? What would be your advice on that?
Yes, thanks, Dr. Trot. So that's a very useful and practical question. Yes, indeed. When we receive the request from our clinical geneticists, so they have the approaches. We have the patient. What kind of experimental therapeutic approach is available? So we analyze this. Usually, we give antisense the priority because it's easy to compare to gene therapy, CRISPR, or the RNA editing. ASO is quite easy to design and rapidly develop, and with the current technology development, it's quite mature technology. And also ASO, the backbone and chemistry, it's very well known. MHR is a regulator. They like it because this is something they know. You just change the sequence. For us, the backbone chemistry are the same. You just change a different sequence. So we usually give antisense, RNA-based antisense the priority. If not, for example, lots of function condition are sometimes RNA antisense is kind of it's not that powerful, and then we change to gene editing or mainly RNA editing or a gene therapy.
May I ask a second question before I open to the floor? Now, you actually told us a very encouraging message towards the end of your lecture that with N of 1 personalized treatment, perhaps using RNA-based therapy, the timeline for the development of a new drug is actually even faster than the traditional way of drug development. It's taking 2 years. Now, that 2 years is actually a very tight window of opportunity. Perhaps for some more serious or severe life-threatening disease. So you need a lot of different stakeholders in order to keep that timeline of 2 years. Can you explain to us who would be the important stakeholders to engage on? Obviously, the clinicians would be the one who started the process because they are seeing the patient and they make the diagnosis. Who will be the other important players, and what are their roles to play in this 2 year time frame?
That's a very good question, Dr. Trin. I would say it takes a village. That's something I learned, a sentence I learned that it takes a village. Um, so with I can go through the ASO development. So it will take scientists analyze the genetic defect and decide. We have the committee and then decide immediately which approach can be used, suitable, amenable for this mutation. And then we need to check if patient's material available. For example, if any uh, for example, skin fibroblasts or blood cell available, so we can check the suitable cell model system. If not, we may need to use CRISPR to make iPS cell line. So this will take a few months. And then then the same time, the lab will need to develop ASO. Once the ASO develop, we need to validate at functional at RNA level, protein level, so that expression level if it work, and also functional level if it work. So we need either the research lab ourselves or maybe some other. We usually check if for a specific gene, if we know there's another research lab available, they have all the system established, we will usually approach them to test the ASO in that system. And then after that, that's about the manufacturing and GLP tox. So the toxicity study will take time. Although the FDA guidance is say 3 months, still 3 months GLP tox, and that part is also expensive. And then GMP manufacturing. So that part, GMP manufacturing is not that difficult. So now it's a quite a lot site for GMP manufacturing, and then MHRA regulator approval. So that's the key steps we work on. And then at the end, who will pay it? So this is the final question. Who will cover the cost? I think this is the final but the key question, and currently is usually from the family charity, but I think this is for many other rare disease. If with people with different income level, do they lose the chance? I think this is the ethic issue. So that's something why, if we have established programs funded by government and or funded by a large foundation charity can benefit all the rare disease patients, not based on the income, if they can pay it, if they can get, raise funding to cover it. So that's another question.
Thanks so much. And Dr. Ma has gathered a lot of questions from interested audience. Wei, maybe we can ask Professor Chow one or two questions before we close the sessions.
Sure, sure. Thank you, Professor Chang and Dr. Zhou. So the first question on the floor is that also regarding the toxicity generated by ASO, for example, the neurotoxicity of by one of the target professor you just mentioned, is there any attempts to use AI maybe to select potential candidates or to design the ASO or generate some alternative approach in order to reduce the toxicity? Is that possible, or is that still under investigation?
That's a very good question. That's exactly the field they are doing now, using AI to predict we have the sequence. You can use AI to predict the toxicity. But developing this algorithm need a lot of data. And then most of the pharmaceutical companies, they don't want to share the data. They only share with the one work, but the toxic one, those failed, you won't get it. So that's UpNet's work that try to push either we share our own negative data, those failed trials, and then we ask, through different channel or resources for those negative data. Actually, here, negative data is more important than positive data.
Thank you. And the second question will be, what will be the preclinical models that you typically use to assess the ASO efficacy and safety? For example, the 3-month GLP toxicity test you just mentioned. Is there any preferred or any recommended models by the guidelines or by your experience?
The 3 month GLP tox, that's in rodents. So it will be in vivo, usually in rats. And so for preclinical talks, we usually use, PBMC, that's the cell line we need to, and as also hepatocyte and renal cell. So that's the three major parts. And the others for functional assessment, we usually use, if we have skin fibroblast from patient and the gene is expressed in skin fibroblast, and this is a very robust model system to use. And then also the iPS cell derived iPS cell model, that's another thing very powerful to use.
Thank you very much, Professor Child. This is really a pleasure to listen to your talk. It's very exciting, and you have had a lot of new data since your presentation in the ESHG in 2025. So for the audience, we should thank Professor Child for her excellent lecture, and thank you very much for your kind attention. Professor Child, thanks a lot for your excellent lecture. We should keep in touch, and we should learn more from you.
Yeah, we keep in touch and hopefully more collaboration in the future. Thank you.